So welcome everybody. If you were looking for Hologic's Innovate session, then congratulations, you made it to the right place. Thanks everyone for joining us at this very early hour, especially if you are from the West Coast and it's even earlier for you. Appreciate you guys being here. My name is Ashley Nenninger. I'm part of the scientific affairs team at Hologic, and I'll be moderating this panel discussion. And this is a panel discussion among laboratory directors, and we are lucky to have a really fantastic group of panelists with us this morning, whom I will introduce shortly. So the title of our session is Emerging from COVID, What is the New Normal for Molecular Testing? And we'll be discussing the transition out of the pandemic, return to routine testing, and the lessons, strategies, and in particular the technologies that labs have gained in the last 2 years that they can take into the future. So we've structured our discussion questions kind of into 3 different parts. First, looking into the recent past in the last couple of years, then moving to the present and maybe some non-SARS testing, and finally looking into the future and where molecular testing is headed. So kind of like a Christmas Carol themed version of past, present, and future, with maybe a much better ghost of the future in this story than in the other. So we've got a lot of great questions to cover, and we'll try to get through them in the hour, but if there is time at the end, we'll cover some questions from the audience if you guys have any. So any comments or questions you guys have, hang on to them till the end and we can probably address them then. So without further ado, I'd like to introduce our panel. So first we have Dr. Megan Crumpler. She is the Laboratory Director of Orange County and Long Beach City Public Health Labs in California. Then we've got Dr. Sanjib Bhattacharyya. He is the Lab Director and Special Deputy Health Commissioner in the City of Milwaukee Health Department in Wisconsin. So let's get started with the first question of the hour. Again, we've structured this into kind of past, present, future. So we'll start with the pandemic and pandemic impact. How did the routine or typical lab testing get affected in the early days for you guys? And then how did that evolve over 2 years?
Yeah, thank you, Ashley, for asking. Good morning, everyone. I'm in a local public health lab, so of course our perspective is different than how we operate, given that we have 2 public health labs in the state of Wisconsin, our state lab of hygiene, and us. But our population not only is a very diverse population, but also challenging in a way that, how we deal with that different vulnerable communities. So I remember one of the, pre-COVID or almost same time, that late February, one of the media talking that we are ready, because we didn't know that the scope of this pandemic and how we are going to be responding to that. So that quickly changed as we realized that within a few weeks. So we got our first COVID, I think, March 13th. And we started with the CDC assays. And of course, very soon we realized that we need to really expand the pre-analytic, analytic, as well as post-analytic pieces, because the diversity of the platform and the technology and all of those things coming out from CDC and FDA, the guidelines are so different. So we have to really navigate through that while maintaining our surveillance and expanding that to the level that we need it to be. So I think within first month or so, we also started working with our academic partner at UW-Madison for getting into the COVID sequencing piece, because that was critically needed. So ONT and ONT Technology, Oxford Nanopore, and Illumina came kind of side by side. And then in terms of getting as the numbers were increasing, we wanted to make sure that our pre-analytic automation, as well as that when Panther came in, it was really a lifesaver, because it really gives us the not only the high throughput, but also the quick turnaround, as well as expanding staff. So we really looked into that as one of our main resources, because the staffing needed to be expanded more than what we traditionally do. So eventually, I think the reporting piece was also critical. We wanted to make sure that not only that different extraction and different specimen types we are dealing with, we are also transmitting information appropriately. So all of those combination of things, I think it's a working with the rapidly evolving, changing situation, as well as staffing, and work with our vendors, partners, and the corporate was critical, because we know that the supply chain issues and initial issues with some of the federal assays that we had been dealing with, but we dealt with that with bringing in diversity of the platform technology and approaching that in a way that we need to serve our community.
What were some of the valuable lessons learned that you took away from the pandemic, or what were some of the unique solutions to challenges that you or your lab came up with?
I wanted to emphasize on a couple of things because of the diversity of the systems and the platforms we are talking about. We really need to look into the inventory control, inventory management of all of this coming in, and where we are, because we know that the supply chain issues and other issues are popping up, but we cannot do it by ourselves. One of the things that we really emphasize that with our city and the county EOC that was brought in and working, coordinating with the state, that how we really working with our communities and the partners, because that's a critical piece, that when you're talking about the pandemic response, it is not just laboratory response. So we wanted to make sure that all aspects of that is being covered. And while laboratory is doing the technical part of it, and of course that's a huge part of it, but it's a coordinated effort that needed to be happening. So the lessons learned is that, critical partnering, making sure your staffing, that's another critical piece. We really needed to expand that to the level that we can offer that. And then bringing in our workforce to the level that they should do. Finally, the data reporting piece, that we had a lot of challenges making sure that not only our lab, but all the communities that we brought in and the point-of-care testing sites who are really fitting into the testing need and the reporting, how they're doing it, and transmission of all the results to the state-wide system was critical. So all of those really helped us, and I hope that moving forward, we can take the benefit of those partnering and the lessons learned for future.
So I'm going to touch a little bit on the unexpected challenges, and as I was preparing for this session, I was talking about kind of and thinking about like what progress we've made in the last 2.5 years. But looking back, we counted, because we had to get ready for our CLIA inspection. We had over 22 verifications for COVID, and so many different tests and sample type and extraction platform and this. So when we compiled them all together, it was like, wow, we did all of this in just 1 year. I think the thing that really surprised all of us and Colleen touched on this a little bit was the shortages and how the word allocation became a dirty word. And I hated hearing that word. Never thought I would hate a word as much, but just how they lingered on for so long and impacted everything. So the plastic shortage, it was tips, it was consumables, it was specimen collection. We're still dealing with some of those shortages still today, and it seems like it's something that really may not go away for a very long time. And so I think that was very unexpected for me. It was like we all thought they were going to just be like short term and turn around, and sure, they're going to start making these things again, but it took much longer than anticipated and was much broader than I think we had ever thought it could be.
So Dr. Bhattacharyya, I think you kind of touched on the staffing challenges, and when it comes to staffing, there were probably some very innovative solutions you guys had to addressing staffing challenges. Like you mentioned, different scheduling and different pay levels and so forth. Could you expand on that a little bit and how you address some staffing challenges?
7 days operation, 14 hours a day was really, really a stretch, especially for a small local public health lab that we really need to respond to the 600,000 population of our city of Milwaukee and 1.5 million for the county. Beyond that, the southeastern Wisconsin, many of the counties are relying on us. So we had to really look into that, how we can expand that. So one thing that we really emphasized is throughout the lab, regardless of if you are a technical staff, whether it's in microbiology, molecular biology, chemistry, every one of them, almost all of them, were cross-trained to do many aspects of the testing. Of course, they cannot do everything, but we really had to do that because we knew that hiring new staff, even working very closely with the staffing agencies, and all of those were taking time. And even when it's taking time, we do not know whom we are getting and how quickly we can train them to do things. So we had really no choice, rather maximizing our own staff. And then when we had the newer or the short-term staff came in, we really engaged them for the pre-analytic. Big thing is looking into the acquisitions, making sure all the demographics are right. So all of those aspects were critical. At the same time, working closely with our human resources, DER, and we really relied on our clinical partners as well. There were times that we had to send some of those to the high-throughput clinical lab to help us out. Again, it was really a coordinated effort, and staffing being our top priorities, we wanted to make sure they are incentivized the way the overtime pay. We are in downtown, so there is a huge parking issue. I mean, as simple as that. But we wanted to make sure that when almost the whole building was empty and we are the only people that working in the lab, so the parking was a great incentive for them during the whole time, that they can come and park in the same building and come to work and be supported. There are a lot of protests going on at that time. I want to make sure that the safety of staff was one of the critical aspects of it. We had to really work with the city attorney's office and the city police to make sure that some of the staff are escorted in and out. So all of those things really aggravated, staff's anxiety while they're dedicated to do public health in the lab. So we wanted to make sure administratively and, from the leadership standpoint, we provide all the support. Of course, technical supports were critically needed as well.
So hopefully something that maybe helped with some of the staffing challenges too was automation with technology. So for this question, I'll go to Dr. Crumpler first. What role did technology or automation play in your laboratory's pandemic response?
So, we too started with manual. I remember being so excited that we were able to quickly write an emergency memo to purchase an Easy One, because that was like the hot instrument. And it is comical now. So we moved to auto extraction, and then the California had a program for public health labs with Perk and Elmer to get us their kind of semi-automated system where each step was automated. You just had to move the plates from one to the other. And we thought that was amazing because we were able to run 94 samples at a time. And then we were kind of slow to get onto Hologic Panther, but we were glad when we moved over to that. We were still needing to aliquot, and we were 7 days a week until about a month ago as well. We're down to 6. But when it moved to the direct load tube, I think my staff cheered because we were able to, really just kind of go back to our normal weekend staffing, which was amazing. But for automation in the future, we now have all these instruments in place. We're working on automating our library prep for whole genome sequencing. We just got up and running with that. So I think, my staff are much more open to using these large automated platforms. They don't seem so scary anymore. And, we will definitely be better prepared with what we've, with the instrumentation that we've been able to get through ELC funding through the pandemic. I just wanted to make one more comment related to our staff. And I think, it sounds like we were all using manual assays in the beginning because we didn't have so much automation because we are not typically, high volume laboratories. But I wanted to go back when I talked about my staff cheering. They were so happy because their hands hurt. And, just that relief and that appreciation, because we're so used to doing kind of, more small volume, we still have a lot of, culture based and, so no one realized like how much stress and pressure that was going to be put on our staff and, work related injuries that come along with having to untwist all those caps. And so I think that's something that we didn't have an appreciation before until we got such a high volume, and being able to put in automation in place to reduce those work-related injuries is really beneficial as well.
Yeah, that's a really good point about keeping your staff healthy physically too. Okay, so again, talking about some non-SARS targets, let's talk about STIs for a few minutes. So STI cases continue to rise pre-pandemic, and I think we're getting ready to see those rates continue to rise even further and maybe more dramatically once we get surveillance in the coming years. And during that time too, STI testing suffered a pretty severe setback. So we'll start with Dr. Bhattacharyya. How are public health departments approaching the challenge of bringing STI testing rates back up?
Well, that's a great question. Probably all our jurisdictions are kind of struggling to address that. But I'll start with a positive note here that We have, in the city of Milwaukee, we have 4 different clinics that we serve other than our main lab. So 2 of them are serving for our STI, TB, family planning, and HIV testing areas. So I mentioned earlier that our high-risk, vulnerable population, they really needed to be supported because all the pre-pandemic and during pandemic, the mental health and being homebound, and that aggravated the risk for the population. And STI being one of the unfortunately the highest rate of one of the highest rate of STIs in our certain jurisdiction, we wanted to make sure that while we are responding to pandemic, that we are also maintaining as best as we can our STI services. So we had to work with our clinic, looking into the clinic workflow, as well as making sure our laboratory testing supports are there. So I think we are fortunate that we worked very closely with our outreach clinic as well as our own clinics, working very closely, maintaining our STI surveillance for all the things we do. That means that CT, GC, TRIG-VAC, MJ, HIV, and HSV, everything else. Challenges we had, of course, primarily with our clinic and getting people to the clinic. So early on, that was a huge challenges, but we have to really do a lot of outreach efforts and making sure that they're being supported. And then people coming to the clinic, that was the initial challenges that we have for several months. And because we continue to have the message out that the service is still available and making sure, so that we have seen some reduced number, but we're able to maintain those for the and clinical laboratories also focused so much on the SARS-CoV-2 testing. We really needed to make sure that we provide those supports they need. We brought in the self-collect and extragenital, which we continue to be expanding as well.
And Dr. Crumpler, same question for you. How is your laboratory or region approaching this challenge of bringing STI testing rates back up?
Sure. So, I mean, even prior to the pandemic, the STI testing world was really interested in increasing access by self-testing, home collection. And so there are numerous mail-in testing programs, though there are some regulatory challenges around self-collection and self-testing for STDs. There's currently only one FDA-approved test to test at home for HIV, so it's a very small field right now. But during the pandemic, telehealth kind of became the norm, and so we needed to figure out how to get these people tested. Obviously, if you attended the plenary yesterday, I talked a little bit about how really SARS has catapulted the self-testing world, and everybody is able to and wants to, easily test at home. If you're not aware, there's a resource by NASTAD and APHL where it's a toolkit for self-testing. So labs and health departments can take a look at that and kind of get a feel for, the different terminology, the self-collection versus self-testing, and, kind of gather some ideas and get some tips and tricks on how to implement these programs. But I think that's really where we're going to see some growth and come out better than we were before in that arena, and help to increase access to STI testing.
If I may add a little more into that, so one of the things that we were able to do that and SARS point of care testing to many of our 15 of our communities, that was very unique. That was actually really helping and complementing the public health lab surveillance. One of the things for what we are trying to do for the STI testing, keeping that relationship as best as we can to those populations that were either underserved or the vulnerable or not addressed appropriately. So beyond SARS-CoV-2, we are looking into potentially doing more rapid molecular point of care for those sites and then bring the reference testing back to our main lab. The reason behind that is, like Megan mentioned, that accessing them to the governmental clinic is sometimes a challenge, but they are more trusted, are more relying on their community small clinic site to go and be tested there, because those are the things that we really normally don't count on or maybe overlooked. So this is an opportunity for us to making sure those populations are served. Eventually, that's cumulatively helping us to maintain or expand our surveillance for the STI and other disease areas as well.
Another sort of or SARS adjacent topic, molecular testing needs for surveillance. So are any of your labs looking to perform any environmental surveillance testing? And for this, it seems wastewater is one of the main topics here. I know for SARS, this has become a hot topic in recent years, and it seems like it could be a very good tool, but potentially a good tool for other targets as well. So
we actually started exploring. We had a partnership with UW Madison, and then Rockefeller Foundation supported pilot study looking into the air surveillance for COVID. So we had been very, very successful, even though in a small scale. Not only, we worked with Thermo Fisher Scientific on one of the sampling devices that they developed. So we were able to look into some of the school system, our shelter sites, and childcare system, as well as looking into the airport and other areas that air sampling followed by extraction to do the SARS-CoV-2 and expanding that to all other respiratory pathogens. We had been successful getting some of the sequencing out of those air surveillance. Very unique opportunity for us. And we have a poster, if you haven't seen it already, that it's virtually being presented. So this is a wonderful opportunity for us to start thinking beyond our routine surveillance and think about a situation that we'll not be able to do that high volume of testing for a long time. So if we have those kind of a system in place that may be really helpful for our air surveillance and getting a prior indication that what's happening in that congregate setting that may need additional testing. So decisions on the testing, what kind of resources we may need, and making sure some kind of a kind of a screening opportunity for us for the air. So beyond the water or wastewater surveillance, I think this is one of the things that we are looking into, putting into our Center of Excellence application, and if it goes through, we'll see how we can expand that opportunity to all the congregate setting that we have the high risk.
What was that poster number? Do you remember? I think it was the lower one. It was on the one side with the lower number. So, okay. So let's move on to finally looking ahead to the future and molecular testing going forward. So for this next question, I'll start with Dr. Bhattacharyya again. And this is back to STIs, to the topic of STIs. So with the incorporation of resistance-guided therapy and CDC's STI guidelines that came out last year, where do you see resistance testing headed for public health laboratories?
Yeah, that's a great question. This is a new frontier. I must say that the paradigm has changed and changing very, very rapidly. And looking into the CDC, not only the CDC guideline has it now, many of us that some of you may be familiar with some of the program, the GIST program that CDC has for the Neisseria gonorrhea, but we are also part of the program called SURGE, Strengthening U.S. Response to Drug-Resistant Gonorrhea. Currently, there are 8 jurisdictions that in the United States who are serving on that, primarily looking into the drug-resistant gonorrhea. But the purpose of that is expanding that resources beyond those jurisdictions that we are already serving. So I think that's a critical need because, as we know, that there are only 4 drugs, or I should say that 4 of the primary drugs that could be used for treating gonorrhea. But not just about the gonorrhea, but Mycoplasma genitalium, that we know the cephalosporin resistance are popping up. There are critical need to look into the Trichomonas vaginalis as well. So I think the resistant-guided therapy is one of our clue or key for the clinicians to look into the STI treatment for the test of cure, disseminated gonococcal infection, and many of the high-risk situation. And for the public health laboratories, which traditionally we did the surveillance for detection, we really need to look into that, expanding that, because we have the tools, we have the resources. It's a matter of working with the right partners, including the corporate partners, as well as academic partners, to bring it on board. Because I think that's how we can really help with our clinicians and treating our patients in a right way. And we know that antibiotic resistance is going to only grow. So not only for the bacterial, but antiviral is also another areas for potential growth. So we can look into those. So I think maximizing the resources we have, working with our CDC partners as well as local jurisdiction and corporate partners will be the way to go to expand on that.
Okay. And jumping ahead a little bit here. I'm going to switch from STI to respiratory. So we've all been testing and talking a lot about SARS and one particular respiratory pathogen in the last couple of years. But in the future, plans for respiratory testing in general are probably going to change. So what are your labs or your region's plans for sort of general respiratory programs going forward, including both testing, surveillance, and sequencing? And for this one, I'll start with Dr. Crumpler.
Sure. So, California is a large state. We have one state public health lab and 28 local public health labs in our network, in addition to our academic partners. And so, prior to the pandemic, we had the state-run respiratory lab network for primarily influenza, but we also track other respiratory viruses through that program. During COVID, our CDPH stood up what we call COVIDNet, which is for variant surveillance and whole genome sequencing for SARS, it was a really amazing accomplishment, kind of modeled on the PulseNet program, where local laboratories, academic laboratories, a state lab, was sequencing and all going into the same, basically the same repository using the Terra workspaces. And so our legislature is really actually excited about this, and we have Senator Pan, who's really active in public health, and so he has a bill that's in the process of moving through the process right now to establish a viral surveillance hub in the state of California, kind of leveraging off what we've developed in COVIDNet and transitioning from like passive surveillance to a more active surveillance program, which is, I think we all learned, is something that we need to do in the future if we're going to get ahead of these pandemics. So it hasn't made it through yet, but we are working on it. Our California Association of Public Health Laboratory Directors supported the bill. I've spoken to the Senate Health Committee to kind of educate them about what's needed in the state of California, and so we're hoping something like this goes through, because it really shows from a legislative standpoint, for them to see the importance of this kind of program in the state, and hopefully they'll fund it.
What steps is your lab taking to be better able to combat the next pandemic?
Sure. So I'm going to focus on some of our informatics initiatives within the laboratory for this one. One of the things that we've just started implementing, which we realized was we really was a setback for us at the beginning of the pandemic, was an inventory management system for all supplies and consumables within the laboratory. That will definitely prepare us because we'll have, if we do run into these shortages, we'll have exact numbers of what's available. We'll be able to find things. We're trying to streamline, down to like 2 types of gloves, and for pipette tips that are used by multiple departments, like consolidating all of those, so we can help eliminate waste, but also keep good inventory of what we need. We are hoping to implement the lab web portal with iConnect. We're working with our LAS vendor on that right now and trying to get that up and started. We are also implementing we use Cerner for our LAS, and so we're working on the molecular lab sequence module to help us better manage our molecular workflow. So it basically works in like a 96-well plate format, and tracks the sample all the way through from extraction to whole genome sequencing. So it'll eliminate a lot of our external Excel spreadsheets for managing this data. So staff are pretty excited about that. At first, it was a little overwhelming, but they really see the benefit there in being able to have a scalable option. Like you mentioned, so you can, you can put 2 samples through, or you can put a whole 96-well plate through. It doesn't really matter, and it doesn't change the workflow at all. So I think that would really help us in tracking our molecular testing in our LIMS.
So addressing combating the next pandemic, I know that every time we had the situation, we talk about that, are we ready, and lessons learned and move forward. But we have seen every pandemic give us a lot of surprises, and of course, this COVID is beyond everything what we have done. A couple of things I will mention about this is looking into the internally our staffing and diversifying the staffing. So we traditionally hired, highly trained, or if we can find the trained individual and put them in the area that what they fit for. But I think we really need to start looking into more of a diverse skill set that we need in the laboratory, number one. Number two, the technology. We all know that, and thanks to ELC and other CDC and other funding that we had, that we really expanded on the technologies, including the next-gen sequencing, which is going to stay with us and which is going to be the future for us. And looking into the high throughput and high-end technology, not only bringing them on board, but also making sure that the performance of those are evaluated, whether there is a clear evaluation or a clear validation needs to be done. Those are critical aspects of it, and how we translate that results to our AP and other partners or the clinicians that need it to be. And number three is another big one is the funding. I think William mentioned about that how we really look into that sudden huge influx of the funding, and if that really dries out, we have a lot of new toys and technologies and also new staff as we expanded, how we deal with that. So we have to be really strategically navigating all of those, making sure we continue to work with our non-traditional partners, including academic and as well as other partners we were able to brought in. I think collectively this should be a different hot wash and learning opportunity for us as and when we exit this, and hopefully that will give us a better direction. I don't think that we have the right direction for the next pandemic yet, but at least the strategic process of mapping those for future already began, and we're hoping to expand that for the next level.
That's great. If there are any burning questions from the audience, maybe we could do one really quick one. Otherwise, we're at about time here. So if you all will join me in thanking our panelists this morning for joining us and for a great discussion. Thank you very much.